So hello students, this is the next class. At previous class we have studied about the bacterial cell signaling and their function, their how they work, etc. Today class will study about quorum sensing. What is this quorum sensing? The cell density depends about the release or stimulate of autoillusion. Here in low density, the cell can trigger um, the response of gene expression due to uh, low concentration of the signals. But in a high density, the cell release more signals and uh, the cell auto induce by triggering itself the threshold by triggering itself after reaching the threshold level. Here the population of bacteria where the gene expression increase and different chemicals are being released to protect themselves or to express more expression they express more or so expression high of high level. What is the example of quorum sensing? In first example is Vibrio fischeri, that is bioluminescence. Here the marine bacteria living in symbiosis with the Hawaii bobtail squid. At high density in the squid, light organ quorum sensing trigger the loss operand. Due to this, uh, the glowing provide counter illuminations camouflage for the squid. But and the second example is Pseudomonas aeruginosa, that is a pathogenicity, an opportunistic human pathogen, wait until release the massive quorum before launching an attack. It results in synchronized release of virulence factor and tissue damage toxin to overwhelm host immunity. Then about biofilm production. What is the biofilm? It is a protective layer that created by the bacteria to protect themselves after being overpolluted in a particular area. What is this definition? The structural community of cell enclosed in a self-produced polymeric matrix high resistance to antibiotic. When during antibiotic attack or antibiotic inductions, the cell initiate or adhere by weak force first, then strong addition leads to microcolony formations. After colony formations, they release some uh, mucus or some protective layer that is known as biofilm that protect from antibiotic, and after the course of antibiotic is uh, finished, they release from that area and spread again in particular organism or or other things. Autoinduction in both in gram negative and gram positive bacteria. In gram negative bacteria, the AHL acyl homoserine lactonase enter through the plasma membrane without any channel. They can travel across the membrane, and the receptor present in cytoplasm. The classical system is Lux R and Lux L. In gram positive, the oligopeptides that cannot be crossed through the membrane. They require a ABC transporter to transport the receptor present in cell membrane require TCS to activate, and the system is a AGR system in S or S. This is the summary. After sensing the different ligand, it activate or move chemotaxis toward the uh, signal. After the um, after chemotaxis, or they know about the antibiotics, something else, they gather and form a microcolony and release some mucus or protective layer to protect themselves from the antibiotic. What is the importance of pathogenicity? Here, uh, signal detect exactly when to deploy virulence. Pathogen use this network to establish colonization. After the colonization, they actually protect themselves from the antibiotic. After that, they release toxin that hijack post-erotic signal. Example, locking G alpha protein, exploring CMP level, causing devastating disease. So this is all about the quorum sensing. You should study more about this. And in next class, will study about uh, interaction, cell to cell interaction, or etc. So at least read that part for better understanding.